Good evening, everyone. This is another opportunity to still meet again in this 2026. This is going to be our second meeting. Remember that yesterday we were able to look at what Mendelian laws were. So we look at the first law, which has to do with the law of dominance, the second law, which has to do with the law of monohybrid inheritance or law of segregation. And we were able to look at what the first filial generation is going to be in case of monohybrid inheritance. Then we also went as well as looking at what is going to be the second filial generation. Also, we also observe, we also examine, and we are able to look at what is going to be the situation, that is a verification of third Mendelian law, which has to do with the law of independent assortment. And don't forget that that time I gave you an example of a kind of dihybrid crossing, which we were able to look at. And later on, I gave you assignments, what I can call a take home that you should do. Few people submitted, and up to now, many people are still yet to submit. This is another opportunity for you because this is still going to be another, probably another task for today. So, without wasting our time, having examined what Mendelian laws are, so today we are going to look at what are the limitations. Don't forget that what we are talking about, Mendel's law, is a law that is, these are set of law that have been formulated in almost 2, 3 decades ago now, and that means there's been a kind of a development in the field of genetics as well as a lot. So we are going to learn what are going to be the limitation and why sometimes Mendelian law might not actually um, be applicable. So today we are going to look at non-Mendelian laws, application of non-Mendelian laws of inheritance. That is, these laws that what are the situation that can warrant Mendelian laws not to be operational or not to come into force. So we are going to look at that as we go on. Okay. Today, what are the first thing we are going to look at? What are the limitation of Mendelian law? Mendelian law, number one, you have to know that one gene will control one trait. If any organism is going to obey Mendelian law, that means it is one gene, one character. One gene, one character. If you are considering the height, that means um, that means one gene is responsible for that, and that is the only way where we can have second law and third law of inheritance coming into play. That is, if you are talking about one gene with a one trait. Then another one is that genes, Mendelian law, genes show complete dominance. You know, I told you the first law was, and the first condition was that the two contrasting characters, one must be dominant and the other one must be recessive. So that is another one. Then another condition of Mendelian law is that the allele must separate independently of the another. So um, the allele, the character, the gene must be able to separate. I told you that they have equal number of chances or the probability of either Y, capital Y and small letter y, or capital letters or small letters. So that means for dominance, you have it must be separate and it must be able to be independent of another. Then on law of Mendel's, the impact of environmental or the environmental impact is so negligible, although it also has some kind of situation whereby we have environmental condition coming into play, like the nutrition, like temperature, like all this. So environmental condition have very little or no effect of on Mendelian law or Mendelian principle. So today, without wasting our time, we are going to look at certain conditions. I think like 5 different conditions whereby the law of Mendel's might not come into play. The first one, without wasting our time, is what is called the incomplete dominance. Incomplete dominance. So, and that is ob obtainable in a situation where you have the red flower, when you cross it with white flower, instead of you to get red or this one. So let's look at this diagram. It's going to help us to understand it. So look at this diagram. Look at this diagram, flower. So red, you can see red flower and you can see white. So the red, that is a capital letter. Red is a homozygote. Homozygote. So homozygote red and heterozygote R, which is white. So when you cross red with what? With white, under normal circumstance, the gamete, capital letter will come from here and small letter will come from here. So instead of producing either red or white in the first filial generation, it is going to produce in pink, which is a heterozygote, that is capital letter and small letter. So it you can see that pink is quite different from either red or white, but it is a combination of white and red. That is why it is pink. So that is a situation, the first situation whereby Mendel's law might not be operational. That is where Mendel's law might not be in operation or might not be applicable. So that is the first condition. So don't forget that I told you yesterday, for us to be able to get second filial generation, what we have to do is we have to crossbreed the product of this first filial generation for us to be able to get second filial generation. If you look at it very well, that is telling you that our first filial generation here is pink. They are pink. Okay, so that is heterozygous. That is capital R, small r. So let us now see what is going to happen. Now, now, under normal circumstance, the first filial generation is represented like this. That is capital by the use of a Punnett square. Punnett square. So this is these are going to be the gametes. Capital letter, and for the Y, which is recessive, small letter. So that when you cross capital with small, we are going to, that is 1 1. 1, we cross it with 1. So you are going to get this. When you cross 2 with 1, you're also going to get pink. When you cross 2 with, 1 with 2, you get heterozygote, which is also pink. So that is a situation where, so the first condition where Mendel's law might not hold is a situation of what? Incomplete dominance. Situation of incomplete dominance. So let us look at the second condition, and the application of this codominance is very helpful for us. Sorry, the application of incomplete dominance is so very helpful for us in plant breeding to produce intermediate what? Traits. So, and it's also very possible for us to in ear texture. So you can see different people with different ear texture. So the second condition we are going to look at where the law of Mendel might not hold is a situation we call codominance. Codominance occurs when both allele are in heterozygous. They are in heterozygous and are fully expressed. They are fully expressed without blending. So, and human ABO blood grouping is a typical example of that. So, individual with AB, that is, so somebody with group O, is either, there are four categories: people with A antigen, people with B antigen. Those that are A antigen, they are classified as A. Those with B antigen, they are classified as B. And those with two combination of A and B, they are classified as AB. And those one with neither A nor B antigen, that is, they do not have any antigen on it, that is blood group O. So let's look at it this way. So you can see those with A allele, those with A allele, you can see the color. That is, I think this is deep blue color. And those one with B, that is a green color. And those one with O, that is no allele at all, recessive. So instead of what happened in Mendel, whereby one allele is going to dominate the other one. So in that situation, it is not applicable here. Two alleles are codominant. That is, neither A, I mean both A and both B are dominating. So whereas O will only serve as a recessive. So when you now look at it this way, an individual with blood group A, that means O. Wherever you see O, that means it is recessive. It is A that is going to manifest. Phenotypically, this person is group A. Phenotypically, this person is group B. So don't mind what you have here, which is O, because O is recessive. So O is going to overshadow, or is going to mask the allele of either A, I mean both A or B. Well, both A and B is going to overshadow it, so that by the time these parents, a parent with blood group A, AO, and a parent with blood group B, if they give birth, that is first filial generation, they are going to have this set of people. AO, that means the person is blood group A. AB, the person is blood type AB, allele of both A and B are present, and blood group AB, that is BO, and they will also have probability of having OO, that is neither A neither B. So they are going to occur in recessive. So one of the ways where, or condition that um Mendelian principle might not hold is blood grouping in human, which is called codominance. The first one is in red and white flower when you cross it together, and there are certain plants that that one condition will take place there. Okay, so the second one is codominance, where instead of one of the allele to be present and the other one will be recessive, we have both allele that are going to be co-dominant. Two of them will be dominating. That means both will be dominating, whereas O, yeah, is going to be recessive. So that is what we have in human. Okay, and that is human blood group. So we can actually see what we are saying here in another very good illustration. So ABO genotype in the offspring. Okay, so don't forget that okay, this is allele, ABO allele inherited from mother. So ABO blood grouping, that's why we call it ABO. Okay, so when you cross two, I mean, one, you are going to have A, that is blood group A. So when you cross B and A together, you have AB. And when you cross A and O together, you have A, because I told you that O is going to be recessive. O is not going to manifest at the end of the day. So when you cross A with B, you have AB. When you cross B with B, you have B. When you cross B with O, you have B. Okay, so going to the allele O, which is recessive now, recessive. So when you cross A with O, it's going to be A. When you cross B with O, it's going to be B. And when you also cross O with O, O is going to manifest. That is going to be in recessive condition. Okay, so that is the second non non-compliance non-compliant with Mendel's law. So, and it is very, very applicable. One of the reasons, nature do everything for certain reason or for certain purpose or for certain advantages. Okay, for certain set of advantages. So it is what we use in blood transfusion compatibility. The fact that this allele one is, both are co-dominating over the recessive one has actually made it very, very possible for us in successful blood transfusion. Okay, so that is that. So the third condition where Mendel's law will not be operational, where Mendel's law will not be applicable, is called multiple allele. Multiple allele exist when more than two allelic form when two or more than two allelic form of a gene occur in a certain population. So the ABO block grouping is still an example of that. So it's an example of codominance, as it's also an example of multiple allele. So that is very, very important. So although an individual carries only two genes, but there are different. In fact, at higher level, we have biochemically, we have more than 100 blood groups or more than 100 alleles that so that is what is called multiple allele. So during that time, Mendel was unable to discover the case of incomplete dominance, the case of multiple allele, and the case of a codominance. He was so ignorant. He did not know that a time was going to come whereby is because he used garden pea plants, that is he used plants. So uh, it was later discovered that not all plants uh, that can obey Mendel's law. There are certain conditions of plants whereby we have only non-Mendel principle coming into play. Okay, so that is condition 3. Condition 4 now is a condition that is called polygenic inheritance. Polygenic inheritance. An example of that is what you have in human: height, the skin color, the level of intelligence, and the body weight. So, and it is a typical continuous variation in human. So this is a condition, polygenic. This is a poly from uh, multiple. Polygenic, that is, we have a single trait like height, but it is controlled by multiple genes. So don't say because you are tall, it is not controlled by only one gene. It is controlled by multiple genes. Skin color, it is controlled by multiple genes. So in a condition like that, it is a polygenic inheritance. The inheritance is going to be in different ways. So we can see what we have here. Although this is more advanced, this is in human genetics. It's more advanced, but you can see the pattern of inheritance where you have ABC, ABC. So you can see when you combine ABC with ABC, that is a capital letter, you are going to see 6. When you combine two two capital letter with this, when you cross it 5 like that, and you have. So, and don't forget, I want you to take note that what we have vertically on the gametes we have vertically are the same sets repeated for what we have horizontally. So, and you should not forget what we did yesterday when I was teaching you about the law of dihybrid inheritance, which is the third law of Mendel. You can see that the gametes, what we have on the horizontal is still the same thing as what we have on the vertical, and you can see. So that is that. So, and this is the distribution of a polygenic. All these will not be able to just know it at this level. For some of you that we actually go further in genetics, in human genetics, you are going to study it more. You are going to learn more about it. But at this level, you should know what polygenic inheritance uh, is and what are the examples of the character or uh, the traits that are controlled by multiple genes. So you can actually go on and learn more about that. So the fifth condition where Mendel's law might not hold or will not even hold is called a pleiotrophy. Pleiotrophy. Pleiotrophy, you can see this is a condition. You can see this is one half of the chromosome, which is a chromatid, and you can see the color here, which is orange. So you can see. So this single gene, this is a gene. The orange is representing a gene on a locus. That gene is located on a specific locus, and out of one gene you can see trait A, trait B, trait C. The one we said the other time was when we have multiple gene, one trait. Multiple gene, that is polygenic. Multiple gene, one trait. But pleiotrophy, that is a single gene, many traits. Single gene, many traits. Okay. So I want you to know that. Okay. Multiple traits, one gene. That is pleiotrophy. But when we are talking of polygenic means one trait, many genes. One trait, many genes is controlled by many genes. But this one is a single gene controlled by more than two characters or two traits. That is a polygenic. Okay, you can see it in another illustration. You can see pleiotrophic gene is the one that is found in blue now. So this gene, which is located on certain part of the chromosome, determines trait A, trait B, and trait C. That is a condition we call pleiotrophy. Pleiotrophy. So please take note of that. And this one is also found in skin color, in eye color, and also in hair color. That is pleiotrophic. So I said one gene influences multiple phenotypic traits. So then the polygenic, a single trait is controlled by many genes. So you should be able to differentiate between. I want you to take um, note of something. The first one is incomplete dominance. The second one is a codominance. Incomplete dominance, codominance. You should know the difference between the two. This one, incomplete, takes place in particular in very common in plants, and codominance human, multiple allele human, polygenic human, and pleiotrophy also human. So the Sixth condition. The sixth condition is what is called epistasis. Epistasis. This occurs in a situation whereby one gene one gene modifies the expression of another on a different locus. So one gene, that means there are two different genes. One gene controls the expression of another one. One gene controls the expression of another one. Okay, so and that is a condition that we see in mice. You can see it here. You see it in mice. You can see when you do when you use Punnett square to be able to do that. So that is that. Okay, so that is these rats. When you cross these rats, that is A C C. That is a gene, two different gene, two different genes that are located on different chromosome. Two different genes that are located on one chromosome. One gene is having influence on another one. So when you multiply it, this is a capital letter A, small letter A, capital letter C, and small letter C. So when you also cross it together, you just discover that this is what you are going to have at the end of the day. Now, when you use Punnett square, don't forget that the second filial generation, what you should know is that the product of second filial generation, that is, which is, the product of dihybrid inheritance, that is 4, 2 raised to power, what, 16, 2n, that is 2 raised to power. So that means the product is going to be 16. For first law, which is monohybrid, the product is 4. For dihybrid, the product is 16. For trihybrid, the product is 4 raised to power 3. It is going to be what? 64. So, but we are not talking about trihybrid now. We are talking about dihybrid. So that is the way, the reason why you have these to be what? 16 products altogether. And don't forget the Mendel's ratio of dihybrid inheritance. It is 9331. 9331. But in a case whereby they give you any example, for example, if they give you any question, maybe in the examination or somewhere, and by the time you are asked to do the crossing, and if at the end of the crossing, that is at the end of your second filial generation, instead of you to get 9331, you are getting a different ratio entirely. For example, there are conditions whereby you can see 4, 4, 4, 4. But what you are going to know is that when you add those ratios together, it must be equal to 16. It must be equal to 16. So now, in this condition now, instead of us to get 9331, what we get is 934. So these four, they are albino. They are albino. You can see that originally in the original crossing, you do not have black rats and you do not have albino. You do not have black, you do not have albino. Then what you have is agouti, which is 9, although it is still the one in so it's present in what? So you can see that all this one are albino rats. We call them albino, and three black, three black and 4 albino, whereas the rest will be what? Agouti. So this is a situation whereby the gene, there has been interaction between the product of two genes. That is, one gene is controlling another one is having influence on another gene, which is not necessarily to be on the same locus. So that is what is called epistasis. Epistasis. So you should know we have that in albino. Once again, the first condition where Mendel's law might not come into play is number one, when we have incomplete dominance. Number two, when we have codominance. Number three is multiple allele. Number four is polygenic genes. Number four is polygenic genes. And number five is pleiotrophy. Pleiotrophy. Polygenic, that is one character controlled by multiple genes. Pleiotrophy, that is you have one gene controlled by, controlling multiple, um, controlling multiple what? Multiple traits. Okay, so that is what you should know. And number six, the sixth condition is called epistasis. Epistasis. Okay, that is that. Then the seventh condition now, the seventh condition is when you now have a gene that is not located inside the nucleus. A gene that is not located inside the nucleus. Don't forget your cell biology. When you have a cell, inside it you have the nucleus, and the chromosome is found inside the nucleus. But there are certain conditions whereby certain genes are found not in the nucleus. So such gene, uh, the pattern of that inheritance is not going to follow the inheritance or according to Mendel inheritance. So that is what is called extracellular inheritance. So some traits are inherited outside the nucleus, mainly through mitochondria or chloroplasts. Don't forget that out of all the organelles, these two organelles stand out. Why are they standing out? Because they also have capability of having certain level of gene also located in them. So they are very, very important. So the key features of this, that usually it is inherited maternally. That is, if such is present in our mothers, that is when that inheritance is going to be extranuclear inheritance. We are going to see some of these when we go on. One of the reasons why we okay, that I don't want you to stress yourself together, more, is just that I want you to look at this diagram. This diagram, it is a diagram that is called pedigree. Pedigree. So we are going to spend our time on pedigree as a different topic entirely. But you should know, wherever you have a wherever you have square in genetics or in pedigree is representing male. And when you have female, that is when you have what? When you have a cycle. Circle is representing female. And if you have male, when you have square unshaded, that means it is not affected. When you have square shaded, that means that male is affected. When you have circle shaded, that means that is female affected. When you have female and when you have circle unseeded, that is non-affected. So you can see more. I have in my other videos, I think last year videos, you'll be able to, when you go through it, you will see where I actually talk deeply on what is called the pedigree. So please, you can see more of my videos on mitochondrial inheritance, or what you can call pedigree, for more explanation on that. So, and this is just a kind of illustration, just to illustrate. So mm, you can see that we are actually dealing with mitochondrial inheritance, and you can see it. Okay, so um, that is that. Now let's now go to. Don't forget that I told you at the beginning of this lecture is that one of the attributes or one of the characteristics of Mendelian inheritance is that environment plays a very negligible effect on it, very negligible effect on it. Whereas in non-Mendelian inheritance, environmental factor play very, very pivotal role on it. So that's why you need to know. And don't forget that this course is actually genetics and the environment. That is, how can you talk about any condition, any genetical condition in that inheritance without talking about the influence, the impact of environment on it? And we actually look at different environmental factors that have different effects on gene or on genetic makeup. Okay, nutrition plays a very pivotal role in non-Mendelian principle. Temperature play a very pivotal role on temperature, light, chemicals, and toxin, and so on and so forth like that. Okay. Now, for your take home for today, I want you to tell me, tell me what are the effects, what are the effects of environmental conditions, the effect on environmental condition on the inheritance of mitochondrial inheritance. That is, you just pick non-Mendelian inheritance principle. I want you to look at pick multiple allele, that is ABO block grouping. What are the effects of environmental conditions, changes in